Hello everyone, welcome back to this video series on clinical trials programming. In this video, we will try to understand the context for cell phenotype findings domain of SDTM.IG 3.4. So we'll try to understand the concept of blood and immune cell. So blood contains different types of cells, each performing vital roles in health and defense. So main types of blood cells include our red blood cells, which carry oxygen throughout the body. And we have white blood cells, so these are part of the immune system and these help us fight infections. And we have platelets which help in stopping bleeding by forming clots. So white blood cells are further categorized into lymphocytes, monocytes and granulocytes. And T cells, B cells and natural killer cells are part of our lymphocytes, and neutrophils, eosinophils and basophils are part of our granulocytes. So these immune cells play distinct roles but often appear similar under a microscope. So how do we classify immune cells using markers? Immune cells are identified and classified using proteins found on the surface of their membranes. So these proteins on the surface of their membranes are called markers. These markers are named CD. CD stands for cluster of differentiation, followed by a number like 3, 4, 8, 19, 56, etc. So the markers are named as CD3, CD4, CD8, CD19, CD56, etc. So cells are often defined using combinations of markers. For example, the cells which have CD3 marker and also CD4 marker, so they are identified as CD3 plus and CD4 plus, and these are called helper T cells. And these helper T cells coordinate the immune response by activating other immune cells. Similarly, we have cytotoxic T cells, and these have CD3 uh, marker positive and CD8 marker positive, and these kill infected or abnormal cells directly, and like our viruses or cancer cells. And we have B cells which will have like CD19 marker positive, and these help make antibodies to fight off germs and remember them for future protection. And we also have something called as natural killer cells. On these natural killer cells, the CD56 marker is positive and CD3 marker is negative. And these helps act quickly to kill infected or cancerous cells. So basic idea is that you are classifying the cells based on the presence or absence of a specific marker. So the classification process is known as cell phenotyping, and it is typically performed using a technique called flow cytometry, and this technique uses labeling of the cells with fluorescent antibodies, and these help us measure both the presence of a particular marker and the level of expression of those markers on each of those cells. And self phenotyping helps us in diagnosing immune disorders, monitoring treatment responses, and also help us understand immune system changes during disease. In addition to identifying cell types, markers also help us determine the cell state, such as whether a cell is naive, activated, memory, or exhausted. So what do we mean by naive? So whether a cell is newly formed, or what do we mean by activated? Whether cell is equipped with all the infrastructure to fight against the infection or cancerous cells, and etc., reflecting its role and stage of the immune response. So why do we capture cell phenotyping data in clinical trials? Immune cells, as discussed, play specialized roles, and subtle changes in their types or behavior can indicate disease progression or treatment effects. In many clinical trials, especially in those of immunology, oncology, or vaccine trials, it is important to track the distribution of specific immune cell types and measure how markers like CD4, CD8, or CD19 change over time or with treatment. And we may be also interested in detecting the activation, suppression, or imbalance in the immune response. So this level of detail goes beyond general blood counts. Though we know that general blood counts like number of white blood cells, number of red blood cells, and number of platelets is captured as part of our laboratory domain. But this level of classification with respect to the markers is not part of our regular lab domain, and these require marker-based techniques which use techniques like flow cytometry to analyze this phenotyping, and that data is stored in cell phenotyping. Now let us try to understand the basic variables that are related to CP domain based on the underlying context we have for CP. So first is the subject, and we have a variable named uSubjD to store the information related to the subject, and the specimen. So we have a variable named CP_spec to store the specimen that is being analyzed, and the method is used, and we have the variable named CP method to store the method that is being used to analyze the phenotyping on cells. And then the cells. So we store the particular cells that we are analyzing, the name of the cells that we are analyzing in the test CD and test variable combination. And then we have now a variable that is used to store the marker related information. So that is CP_MRK_STR, marker string. And then the result of the assessment is stored in the series of variables related to our result and result unit. CPORRES, CPORRESU, and U is for unit. And we have our standard result variables available to store STRESN, STRESC, and STRESU. And then the limits of the lower limit allowed and upper limit of the normal range are stored in CPORNR low and CPORNR high. And if we are standardizing the units, those limits go into CPSTNR low and CPSTNR high. And we also capture the normal range in the variable named CP, normal range indicator, whether based on the comparison of our result with our normal range low and high, the result may appear like low or high or in between the limits. So that information of low, normal, or high is stored in the variable CP and RIND. So these are the basic minimum variables that are needed for a CP domain. So when compared to other findings domain, the additional variable that we have here is the one which is related to the marker CP_MRK_STR, marker string. We have a few other variables which are very specific to the CP domain, so but those are not regularly needed. So thank you for watching and keep learning.